Okay, let's keep going. So we talk about the water has high heat capacity, surface tension, and this because the hydrogen bond to keep it together. And I call this perfect glue. This is not a super glue. This is not very strong, so we can break it. And in the biological system, we also use hydrogen bond to keep the molecule together because this is a glue, like the sticky note. You can put it back, you can take it off. So we can break it if we need to. So far, we talked about three different kinds of bonds: ionic bond, electron giving away, covalent bond, electron sharing. And we talk about the hydrogen bond, that's the weak bond, that's the intermolecule. These two are intramolecule bond. Okay, now let's look at the solution. So you take the solvent are the substance, like the sugar, the salt, you put them into usually water, and that's the solvent. Then you create a solution. And most of them, we're going to put those molecules into water. So we have water solution. And this molecule, if they can dissolve in water, we call it hydrophilic. Like the sugar, the salt, all these are hydrophilic molecule, substance, because they can easily dissolve in water. And why they can dissolve in water? Because, well, water is polar. And when you put salt into water, it becomes the ions, and you can, like the sodium ion, you can use the negative side of the water surrounded. So you create a hydration shell, so make it very happy. But for the molecule, they don't dissolve in water. We call the hydrophobic molecule, like oil. So we know the oil and water don't get along well. So they're gonna form a layer and stay together, and that's the hydrophobic. Now let's look at the cell membrane. Cell membrane is made of phospholipid, and we talk about this molecule. This molecule just tends to have a head like water, and the tail don't like water, so it's called the amphipathic. And you need to use this molecule to make the cell membrane, and now you are facing a challenge. The challenge is in the cell, the inside, the outside are all water. And you have the head like water, okay, this is not a problem, but the tails, they don't like water. So it turned out they need to create a double membrane. They need to take another layer and make the head part, they like water, facing outside. And the tail, they don't like water, they put them inside. So all your body cells, the cell membrane is double membrane. It's double membrane. Now let's look at the solution. And we use the blood to explain it. So your blood, you have blood cell and plasma. Plasma is mainly water with salt and some protein. So when you get the immune shot, those antibodies, they are protein, they stay in the plasma as well. So we can put them into different categories. If you have the plasma only and with salt dissolving it, and those substance molecules are super small, we call it solution. So the plasma with no protein, and that's the solution. And if you add antibody into it, protein in water, this plasma we call it colloid. So you have the substance a little bit bigger. So plasma is colloid. If I ask you the plasma with antibody, and this colloid. And now we add blood cells in. Blood cells are actually big, and we call the suspension. So if I ask you, okay, the blood is actually a suspension because they have a big molecule, and which is the blood cell. The blood cell is 99.9% red blood cell, so they are big suspension. So we have three different kinds of solution. And unique one, emulsion. Emulsion is you have water, you have oil. And we know they don't get along well, but for some reason, well, this solution can mix good enough. And good example is breast milk. So in the breast milk, you have water, you also have oil. And when you freeze the breast milk, they separate. So you found those freezed breast milk, they actually have two layers. And those oil, don't throw it away. These are actually very good oil. These are high omega-3 oil, very good for the baby's brain to develop. So don't throw this oil away. These are good oil. And in the breast milk, they're mixed, so it's called the emulsion. And the solution, sometimes the molecules you put into the solution, they can dissociate and they produce a lot of H plus hydrogen, and we call it acid. So the acidic solution is the one with a lot of H plus. So you have the strong acid, weak acid, depends on how much H plus you have, how much hydrogen you have. And let's look at the base. So you have acid, you have the bases. And the basis is two definitions. One is you have a lot of OH. So I found the old textbook, 10 years old textbook, they'll tell you base is you have a lot of OH. And the new textbook definition is they can accept more hydrogen. And both of them are correct, because when you have a lot of OH, and they can accept more H plus, because OH and H plus together they become H2O, so they can remove more H plus. So both of them are correct definition. Either they accept more H plus or they have more OH. So they can have a strong base, they can take more H plus away, or a weak base they can take less H plus away. And when you add acid, base together, acid, very strong acid, hydrochloric acid, and you add the base, what can happen is those H plus from the acid, OH from the base, together they become H2O. H2O is water. And the rest of them is potassium and chloride. Potassium chloride together become a salt. So when you add acid and base together, you become salt and water. This process is called a neutralization. And a way to measure how much H plus you have in the solution is pH. So the definition of pH is negative log of H plus, of the high concentration of hydrogen. So it's a log scale. It's a log scale. And depends on how much H plus you have. Because it's a log scale, so every time you change the number, it gives you a number from 0 to 14 based on the concentration. And it's a negative, so because it's a negative, so turn out the number is smaller, you actually have more H plus. So the smaller the number is more acidic, because they have more H plus. And second is a log scale. It's a log scale, so every time you change number by 1, like 1 to 2, 2 to 3, you don't change one time, you actually change 10 times. So if I give you two solutions, one pH 1, the other pH 2, I ask you how many times more H plus in solution 1 than solution 2, it turns out it's not one time, it's 10 times. So compare with pH 1 solution with pH 3, is 10 multiplies 10 is 100. So every time you change the scale, it's 10. So compare solution 1 with solution 4, pH 1, pH 4, is how many times more H plus you have, is 10 multiplies 10 multiplies 10 is 1,000. So it's a log scale. And that's the pH scale. So it gives you a number, tell you how acidic the solution is. So it's from 0 to 14, and it's a log scale. So if number is 0, means 10 to 0 times. So the pH is 0. This is super acidic. And 0.1 m, this is 10 to minus 1. So in the pH scale, after the log is 1. So the smaller the number, is actually more acidic. And 7 means neutral. So like pure water is neutral. And when the number is bigger than 7, it's a basic solution. And the bigger the number, more basic, because they have less H plus. And one thing I want to show you here, human blood. Human blood pH is 7.4, so your blood is actually not neutral. Your blood is a little bit basic. And your body need to maintain homeostasis of the pH of your blood. So this pH is well maintained. So you can only fluctuate from 7.35 to 7.45, very small fluctuation scale. And that's your normal pH, blood pH. And if you like to drink soda, soda is very acidic actually. All the soda you like to drink, they put CO2, they push CO2 into the sugar water. That's how they make soda. So those CO2 dissolving water become carbonic acid, and that's why it's acidic. So all the soda pH between 2.5 to 3.2. So all the soda are very acidic. And your blood pH is 7.4, so your blood is not neutral. It's a little bit basic. And you have a very small wiggle room, so you need to maintain homeostasis of the blood pH from 7.35 to 7.45. That's the normal pH. So if I ask you what's your blood pH, you can give me the range from 7.35 to 7.45. I will call it correct. The easiest way to say is to say 7.4. That's the normal blood pH. And a strong and weak acid depends on how much hydrogen they can produce. So if they can produce a lot of H+, then that's a strong acid, like hydrochloric acid. And pH will be very small, like between 1 to 2. And if they release less H+, like carbonic acid, is a weak acid. And you also have the strong base, the weak base. So strong base is they can bind more H+, or you say they can release more OH. Both of them are correct. And or the weak base, they can take less H+. Because your body pH is so important, need to be maintained, your blood pH need to be maintained, you need something to maintain, to minimize the change. Because every time you breathe, your CO2 level change, or you eat a lot of acid, you drink soda, well, this is going to affect your pH. And why your body pH won't change? That's because, well, your body has buffer. Your blood is a buffer. So what's buffer? Buffer can minimize the change of the pH. So you add acid, it's supposed to produce a lot of H+. In the buffer, well, it becomes a weak acid because buffer take a lot of H+ away. And you put a strong base, a lot of OH into a buffer, it turns out, well, not so much OH left, and because it takes a lot of OH away. So buffer can help you to maintain pH. So that's basically what I said. So you put 3 H+ into the solution. Well, we're going to have 3 H+, make the solution acidic. But it turns out the buffer going to take 2 H+ away, so you only have 1 left. And that's the buffer situation, and that's the non-buffer situation. So compare the non-buffer with buffer. You add H+ into it, and the buffer, it ends up with much less free H+ left. So the pH won't drop that much compared with the non-buffer solution. So buffer can help you to maintain pH. So in your body, buffer is very important because you want your blood maintained in the normal pH from 7.35 to 7.45. If you lower than 7.35, we call it acidosis. It can be caused by metabolic problems or respiratory problem. Your breathing rate is too slow. You can cause acidosis, respiratory acidosis. Or you can cause alkalosis. Your body pH is too high, and both of them you are sick. And when you go out of this range, apparently you die. So your body has buffer to maintain pH, and buffer is very important. Okay, that's it.